Hey, micro friends, welcome to another episode of Let's Talk Micro. As always, I'm so happy that you're here with me. And if this is the first time listening to Let's Talk Micro, well, thank you for choosing it as one of your podcasts. Okay, so I know that in social media and the news, you have seen so much information about the Andes virus and the Hantavirus and the cruise ship. So I thought it would be a good time to make an episode about this. So this episode is the first one of a two-part series where we talk about hantaviruses. And in this one, we do like an overview. We talk about what type of viruses are hantaviruses. We give that explanation. We talk about transmission. We talk about diseases. And then in the second episode, which is going to be next week, we talk about the Andes virus and the whole cruise ship situation. So these are two great episodes, very educational, very informative. So I hope you enjoy them. So let's go ahead and listen to the first one. Hey, micro friends, if you are in the microbiology infectious diseases community, and even if you're not part of it and you're going through social media or you see the news, I'm sure that you have heard about the cruise ship and the hantavirus, and you see all this posts, all this information. As I always said, you get so much on social media. You can use it for real good things, and at the same time, there can be like an overload. So I thought it would be a good time to do an episode about it. So with me today, I have two guests. One of them, he's been a guest before, and then the other one is joining us for the first time. So with me, I have Dr. Ryan Relich and Dr. Stephanie Seifert. Everyone, welcome to Let's Talk Micro.
Thanks for having us.
Yes, thank you so much.
My pleasure. So I thought it will, Ryan. It's been a while since you've been in the podcast. So for those new listeners, and then you, Dr. Seifert, maybe if you can start with a quick introduction about what you do, and anything else that you want to share with the listeners.
Absolutely. So we'll go ahead and start with Dr. Seifert.
Hi, I'm Dr. Stephanie Seifert. I am an assistant professor at the Paul G. Allen School for Global Health at Washington State University. My research focuses on understanding why certain viruses jump between species, especially zoonotic viruses that jump to humans from wild animals or livestock. And so we, in my lab, combine field ecology, evolutionary biology, virology, and computational modeling to study zoonotic viruses and how they make these jumps from animals into humans.
Thank you. And of course, you know me, Luis. My name is Ryan Relich. I am a professor of clinical pathology and laboratory medicine at the Indiana University School of Medicine in Indianapolis. And for Indiana University Health, I serve as the medical director of the Division of Clinical Microbiology, medical director of the IU Health Donor Screening Laboratory, medical director of the IU Health Special Pathogens Unit Laboratory, associate medical director of the Division of Molecular Pathology, and director of the CPEP Fellowship Program. In addition to that, I also have an academic research laboratory. I too study viruses, emerging viruses, in the context of diagnostics development, antiviral countermeasure development, and also a little bit of virus ecology.
Well, welcome back, and and thank you both for joining me today. So for the listeners, we always hear in Let's Talk Micro, we before we talked about the cruise and the virus, and we like to start with some basics. So first, can you start with what exactly are the hantaviruses?
So hantaviruses are RNA viruses, meaning their genome is comprised of RNA instead of DNA. They're single-stranded and segmented. So you might have heard of, say, reassortments with influenza virus. These viruses are similar in their genomic architecture, but they have fewer genome segments. So they're actually kind of a simple virus in terms of their genomic architecture. Other viruses in this group include things like Hantavirus, which is a major public health problem in Eurasia. And then there's Sol virus, which people might have heard of. I think a few years ago, there was an introduction of Sol virus with rats in Washington, D.C. That's happened a few times to the U.S. and also around the world because rats are cosmopolitan and they move around quite freely. So that's the primary host. Those viruses seem to move around. And then they also include viruses that we associate with the New World. And so in the New World, we have viruses like Sonomre virus in the United States, which people might know from the Four Corners outbreak in 1993. And so in the US, when we talk about hantaviruses, we're usually talking about that specific hantavirus. In South America, there are also diverse hantaviruses that are also associated with this hantavirus pulmonary syndrome, like Andes virus, which everyone has heard about now. I think Ryan can probably talk a little bit more about the specific diseases because we do have some geographic structuring with the associated diseases for these viruses.
Yeah, absolutely. Thank you very much. Um, in North America, in the Western Hemisphere in general, as Dr. Seifert mentioned, there are a number of orthohantaviruses found here. It's sort of the big one in North America that gets the most press, of course, is Sonomre virus and its association with a disease called Hantavirus pulmonary syndrome or Hantavirus cardiopulmonary syndrome, whichever way you want to call it is acceptable. There are a couple of other species of New World orthohantavirus from the United States and North America in general that are associated with HCPS or HPS. One's called Black Creek Canal virus. There's Bayou virus. There's variants of Sonomre virus like New York virus and Monongahela virus. There's also, I think it's important to mention too, there are also some orthohantaviruses that have as of yet undefined human pathogenicity, like Prospect Hill virus, which was first found in Maryland. That's carried by meadow voles. And so there's, this huge spread of viruses in Central and South America. There's Laguna Negra virus, there's Andes virus, et cetera, et cetera. But one common attribute of the disease phenotype of those viruses is cardiopulmonary disease. Whereas viruses, orthohantaviruses that are endemic or native to the Eastern Hemisphere are associated with disease process called hemorrhagic fever with renal syndrome. And so, real quick, too, as an aside, there is an orthohantavirus that is found in the United States, in the Western Hemisphere, a lot of different places, and all throughout the Eastern Hemisphere, too. That is reservoired by brown rats or possibly some other rodents as well. It's called Sol virus, which causes HFRS. So there's two different varieties of orthohantaviruses that you can find here in the Western Hemisphere. So Dr. Seifert alluded to the fact that there was an outbreak in Washington, DC, as well as individuals with pet rats that occasionally will develop hantaviral disease associated with those animals. But a little bit more about the disease processes in themselves. So HPS or HCPS is a tripartite or a triphase disease characterized by a flu-like prodromal phase, which you also see with HFRS. But with HPS or HCPS, there's also what's referred to as the cardiopulmonary phase. So in that phase, you see more advanced symptomatology, including pulmonary edema, cardiogenic shocks, shortness of breath, respiratory failure, and in severe cases and fatal cases, of course, death will be associated with it. However, in about, what would you say, Steph, about 60-ish% of people, there's a recovery phase, so they end up getting better. And with HFRS, there's that same flu-like prodrome characterized by a febrile, nonspecific clinical picture with headache, myalgias, arthralgias, nausea. People can develop a flushed appearance, have conjunctivitis. They may or may not have a rash. As the disease progresses, you'll see hypotensive state. And a feature of both HPS and HFRS is really vascular leak. So a lot of the disease symptomatology and pathogenesis that you see is actually associated with increased permeability of microvasculature, whether that be within the kidneys and within the lower abdomen or within the thorax. For example, the microvasculature of the lung is typically the site for most of the disease pathology with hantavirus cardiopulmonary syndrome, or HPS. But in both HFRS and in HPS, you see sort of profound vascular permeability leading to vascular leakage. And then, for example, in the case of the lungs, you see just profound intraalveolar edema. And on histopathologic examination of autopsy tissues from the lungs, you'll see not only viral antigen present within the endothelial cells of that microvasculature, but you'll just see this abundance of fluid collection within the lungs. And in cases of HFRS, you'll see acute kidney failure, kidney damage, fluid overload. But in both recovery phases for both HPS or HCPS and with HFRS, you note that vascular permeability is reversible. And so over time, you then stop seeing that. But another thing to mention, too, I suppose, is the hemorrhagic fever with renal syndrome. In Europe, especially, you see sort of a mild form of that called nephropathia epidemica that's typically associated with pulmonary, which is carried by bank voles. You'll see that in Finland and other places as well. But that disease is generally a bit more mild than like Hantan or Dubrava Belgrade virus associated HFRS. Did I miss anything, Steph?
So we can maybe talk a little bit about, the global distribution and diversity of these viruses really reflects also the global distribution and diversity of rodents. And so we have these phylogenetic groupings. We talk about Old World and New World hantaviruses separately. We often talk about those as associated with the pathogenesis, so HFRS or HPS. It's important to note that the HPS viruses are kind of nested within the diversity of what we call the Old World hantaviruses. And I struggle with this a little bit because if we want to name those after Old World, New World, it doesn't quite make sense. It doesn't quite partition that way because we have, viruses that cluster more closely with the HFRS viruses in the US. And so that would include like crossfactyl virus, which is full associated here and seems to be more closely related to things like pulmonary than it is to SNV. And so it's not totally separated by Old World, New World, but it's also not totally separated by pathogenesis because, as Dr. Relich said, you don't always have, you don't always know whether that virus is going to cause disease in humans. So if we're describing these viruses instead by their pathology or their pathogenesis in humans, so a lot of these viruses we don't know. They're uncharacterized in that way, or we haven't seen disease in humans. So generally, it's easier for us to find viruses if they cause disease because then we start looking for them, what's causing that disease. But we do have also a lot of viruses that we just kind of happened upon. And some of those there's hantaviruses in bats in Africa. There's hantaviruses in bats in South America as well. There's, hantaviruses have been identified in shrews. And so we think that the diversity of hantaviruses kind of emerges out of Eurasia. And likely these viruses cross the Bering Strait with rodents. And then the Hantavirus pulmonary associated viruses, they really originated here.
And before we move on, by now, so you have mentioned rodents and rats, and those are the when it comes to these viruses, those are the primary vectors. Or is there any other animals or?
I would say that, we certainly find hantaviruses in bats and shrews, like I mentioned, but we don't have good evidence to support, I think, that those are the primary kind of reservoirs that lead to human or transmission to humans from blood-like reservoirs. I think one of the kind of open areas of research right now is how diverse the host system is for many of these hantaviruses. Historically, we kind of have this dogma that it's one host, one virus, that, for instance, synovial virus is only Paramiscus maniculatus. And the picture, I think, has evolved since then. It's becoming much more apparent that there are many susceptible different rodent taxa. Here in Washington State, we've just sequenced the virus out of the lung tissue of of voles with Microtus montanus. And so that was surprising for us to have it be that divergent. Then we spoke with our colleague in New Mexico, Dr. Stephen Bradfield, and he's recovering infectious virus out of the salivary glands of many different rodent taxa. And so I think the host picture is maybe more complicated than we had originally thought that it might be, the scope of susceptible hosts and what that means for onward transmission and maintenance of this virus in rodent populations. I don't think we've fully explored. So I think that's a really active area of research. We often talk about rodent excrement as being a source of hantavirus, and as Ryan was talking about these differences in the tropism between different tissue types, between these different groups of viruses, the HFRS viruses tend to be more associated with the kidneys. And so we find them from the kidneys, and they seem to be more readily transmissible. So Hantavirus was a major public health problem in Asia and still is in rural communities primarily, but we're talking tens of thousands of cases per year, or even hundreds of thousands of cases per year, versus a virus like Sonombre, where we see maybe 900 cases since 1993 in the United States. Part of that might be attributed to diagnostic capacity, that I don't think we generally test for hantavirus unless someone's progressing to severe disease. It's likely underdiagnosed. But I still don't think that these hantaviruses that are less associated with the kidneys or renal system in rodents are as transmissible as the viruses that we are most concerned about in the Americas, which includes, I think, Andes and Sonombre. For these viruses, we often find there's an acute phase of infection on the rodent where you can find the virus everywhere, right? They get infected, it disseminates throughout the body. We can find it in the brown fat. We can find it in the kidneys. They're urinating the virus out. We can detect it in the urine. And then they kind of turn on this regulatory T cell response that kind of controls the infection, but it doesn't clear it. And so then these rodents are persistently infected. And in Europe, in Asia, you'll find that those rodents are just always shedding the virus in the urine, even after the acute infection. In the Americas, where we have Andes and Sonombre, we're not totally sure, but it seems like it's more localized into the salivary glands. And so we'll see it instead in the saliva and oral swabs, and maybe we won't find it in urine. So when we're doing surveillance, we're actually looking primarily in the lung tissue, and it's much more reliable for us to detect this virus like Sonombre virus in the lung tissue of rodents than it is in urine or bladder tissue. So with that, I think that, we talk about urine as a concern. And so if you're cleaning up an outbuilding and you start sweeping in, the dry Western US, then you're likely to produce aerosols. So if there's infectious virus present, as you're sweeping, it's possible that you could aerosolize that virus and then inhale contaminated excreta while you're cleaning. And so that's one of our major concerns. And we see people, cleaning out buildings with leaf blowers. That's an aerosol generating procedure. We see people sweeping in outbuildings when they find rodent waste. But we also have, I think, concerns with, like sometimes you go and you turn on your car in the winter and you turn on the heater and you smell rodents and you find them inside your air filter. And this is personal experience. Because these rodents are really hard to exclude from our housing, from our outbuildings, from if you park the car outside. They're seeking shelter, and we create a warm, toasty, safe environment with few predators. So these rodents are really hard to kind of completely exclude from our environment, and we have to be aware that they exist. And it's not necessarily just, they're not keeping their environment clean or something like that. Like, yes, clutter can contribute to rodents, a rodent population in an area, but it's also really challenging. Even new housing, if you built new housing in, a suburban area, but it's new, then it's likely disrupting that rodent community at that local site, which means that those rodents are going to find somewhere else to be, and it might be your house, right? And that's just an issue that we have to deal with. So in terms of transmission, the best way to protect yourself is that if you encounter any rodent excreta or evidence that rodents have been at a location, you need to clean up to pursue something that we call a wet cleanup. And that reduces the probability that you're going to produce aerosols. And so if you put paper towels down or you use wet wipes, something like that, but you're not sweeping, you're not using a leaf blower, and just trying to be aware that there's potentially hantavirus and rodent excreta, not just feces, but urine for sure. That's the best way to prevent that transmission. So it's a persistent problem. These rodents and viruses have been around for a very long time. And I think that's also true from when we see the diversity of these viruses, right? We've been living with hantaviruses for a very long time, historically. And so they're here. It's a persistent problem. We talk about it as a rare disease, which in epidemiology kind of means more like low incidence. But it's also predictable. Like this is a recurrent public health problem that's not going away. And we've also talked, I know there's been a lot in the news recently too about climate change and how climate change might be driving different new distributions for rodents, et cetera, or changing rodent population dynamics. And that's very likely to be the case. The mechanism of how that leads to increased zoonotic risk for us, I think, is something that we still need to do the research on. I will say in our area, it's not just the kind of simple trophic cascade of more rain means more vegetation, means more rodents, means more hanta. I think it's also about the stability of those rodent populations, and that we have tied back to how long the breeding season is. So when you have a milder winter, rodents are breeding for a much longer window of time in that year, and we end up with more rodents and then more contact. So it's not just like stochastic, here's rain, here's rodents. It's we have really high survival over the winter. And so starting out, we've got a bigger population. And by later in the year, we're going to have potentially more hantavirus cases. There's also that kind of like delay to that effect for transmission. So it's definitely still something that I think we should be studying for exactly how people get exposed, but also how rodents maintain hantavirus in those populations. And I think, Ryan was describing the disease in humans, and the early states are kind of non-specific. And we're talking about rural communities that also don't have the same access to healthcare resources. And so if someone has flu-like symptoms here in our area, unless it progresses to severe disease, they're probably not sending out for hantavirus diagnostic testing. And so I think that also really matters in our understanding of the epidemiology for hantaviruses.
Yeah, and even in cases where there are extensive medical evaluations of patients in very high, resource-rich areas, sometimes it's just not on the differential for the clinicians. There can be, quasi-protean presentations of orthohantaviral diseases that, Steph, weren't you telling me a while ago that you had a patient that had encephalitic disease?
Yeah, there's a couple of patients here in Washington State where two people were presenting with cognitive impairment, motor skills impairment, and slurred speech, severe headaches, but no pulmonary involvement. And then they were tested for a battery of other diseases, like Lyme disease. I think hantavirus was maybe the 40th thing that they tested these individuals for before they got a positive test. And so I think that's a real challenge is we also don't, I think, understand the breadth of clinical science that can be attributed to hantavirus disease. I think there's a paper showing maybe 6% neuroinvasive signs for Andes virus as well.
Yeah, and even in fatal cases, not every patient gets an autopsy. Medical autopsies are generally done less frequently than forensic cases are. And so families can elect to not have an autopsy of the person post-mortem. And so in those cases, it's especially difficult to establish a diagnosis because, the spleen tissue, liver tissue, heart tissue, that's all crucial for detection of viral antigen, detection of nucleic acids, things of that nature. And so we don't always get a cause of death whenever a person with various disease processes, probably including pulmonary syndrome or even HFRS. And so some of these things just fly under the radar, and we just don't know the true incidence and prevalence of the disease. And so that's another consideration I think that needs to be taken into account is even in areas with very robust healthcare resources, we may miss it.
So if you're, and this is just something that if you were maybe like looking it up and you see the term, like hantavirus, and you mentioned also orthohantavirus, can you maybe clarify that for the audience? Why the different names?
So I'm kind of an armchair taxonomist. I love names quite a bit. Nomenclature and taxonomy are some of my favorite things, but I'm not a professional. Yeah, so the International Committee on Taxonomy of Viruses is the international subject matter expertise body that defines sort of the internationally used nomenclature for viruses. And so there are common names of viruses, and we'll use RSV as an example, respiratory syncytial virus. That's one I think that all your listeners have heard of and have experience with perhaps firsthand. So RSV is the common name of an orthopneumovirus. That's the genus of the virus. And in the case of hantaviruses, the genus is orthohantavirus. And orthohantavirus andesensi is the species name. So it's kind of, non-bacterial, non-fungal, animal plant taxonomy and nomenclature here. It's sort of non-standard to the first-time observer. But for example, Andes virus is its full genus and species name is Orthohantavirus andesensi. So you get the repeats of the orthohantavirus in the species epithet as well. It's like that with all viruses that have formal binomial nomenclature. But hantavirus is just sort of a generic word that's used to describe these viruses that are categorized within the genus Orthohantavirus. And there's currently 37, according to the International Committee on Taxonomy of Viruses, 37 species of orthohantavirus. But if you call it an orthohantavirus or a hantavirus, I think people are going to know what you're talking about.
It's a new change to binomial nomenclature for viruses. So for some taxa, it really helped to make that change. So the genus for Ebola viruses previously was Ebola virus, one word. And so you might be talking about Ebola virus aerensi or Ebola virus Windibujo. And now it's ortho-Ebola virus, which is very helpful because the common name for ortho-Ebola virus aerensi was just Ebola virus with a space in it. So in some cases, the binomial nomenclature has really helped. And for hantaviruses, I'm not sure it made a huge difference, but it's at least consistent across viruses where we have that like ortho designation for genera. So you can say either, we know what you're talking about. They're not different things. They're just a slightly different level of correctness, I guess. We discovered two more. And Sager quick virus is one from Curtis that we were in the 2025 new species for RNA viruses listing from ICTV. So
Nice. That's awesome. Yeah, and Luis, the way I sometimes teach this whenever I'm talking to people who have for the first time heard some of this stuff is I use, I'll anthropomorphize. I'll try to relate it to. So Luis Steph and Ryan are all Homo sapiens. We're all the same genus. We're all the same species, but our common names are Ryan, Steph, and Luis. So
That's fair, yeah.
we're all kind of variants of the same species here. So, but yeah, that's our common name.
Well, thank you. That's a great way to think of it. So, and Dr. Seifert, you touched on this already, and so when it comes to the orthohantaviruses, and you mentioned that things like if you are like using leaf blowers, or you get those aerosols. What else can you tell us about like ecological and human factors that can drive the spillover from rodents into people?
I think that that's been a lot less studied. We do know for deer mice in particular that we have they really prefer and do very well around agricultural settings. And so they're frequently found around outbuildings. There's a lot of, protection from severe weather. There's protection from predators and outbuildings for those rodents, and they tolerate human presence very well. So outbuildings are a big one for attracting rodents, but also agriculture. So in a place like Washington State University, we're in Eastern Washington, in an area called the Palouse, which is an important hub for production of wheat and canola and garbanzo and a lot of grains here, grain production here. And Peromyscus are granivores, like they love this stuff. So when we build houses adjacent to these fields, we have a constant food source, and then we also have now produced a structure that keeps a stable environment during inclement weather, and then also provides shelter from predators. And so we kind of created the perfect habitat for Peromyscus. It just so happens that they're also adapt reservoirs for Hantavirus virus that don't experience severe disease when they themselves are infected. And so I think it's really trying to keep those animals out of our homes is the biggest thing, but also being aware that when we're going into like an outbuilding, where it's very hard to prevent those rodents from using outbuildings, that we just have to be, cautious and aware that there could be hantavirus present in the excreta. In terms of like environmental drivers, so the 1993 outbreak in Four Corners for Hantavirus was preceded by an El Nino event. And so it was unusual weather patterns that led to a lot of vegetation and growth. I think in terms of being able to do a larger study with our viruses, because there's so few cases tracked, it's actually quite hard to get at those environmental drivers because the incidence is still very low. And so the probability of any one person getting infected is essentially very low. But we know every year that there will be HPS cases. So we've never had a year since 1993 in the United States with 0 cases of hantavirus pneumonia. This year has been particularly bad in the Western United States, and I think that's largely attributable to not necessarily a lot of rain, but instead that we've had a very mild winter in the West. So almost no snowpack. And I think that we've had really good survival over the winter of these rodent populations, and that's led to a larger rodent population and then more readily transmitted hantaviruses. With the hantavirus pulmonary syndrome associated hantaviruses, there's a lot of discussion about how important male-male aggression is during breeding season. So a lot of these rodents aren't necessarily territorial in that they'll like find other males and actively attack them, but they do kind of develop some of those patterns during the breeding season. And so there are several studies with hantaviruses to show a higher prevalence of SMB, in particular in male Peromyscus. And so we kind of hypothesize that that's the cause is male-male aggression leading to higher transmission among males, but it could also be that males are exploring a larger area or encountering the virus in other ways. I think there's still a lot to be done on the hantaviruses. It's a system I think that's less studied than some other pathogen systems for sure. We just kind of assumed that we understood everything about it. We're like, it's deer mice, it's El Nino, and now we know everything. And it's the more that we actually investigate the prevalence, especially across different taxa, the more we're really learning. So it's still even an argument sometimes with people to say that, when you have a chipmunk or a microtus that's infected with pulmonary virus, that they aren't just a kind of dead end host. But when it's 50% prevalence in our microtus and we're able to get the virus out of, for us, sequencing out of the lung tissue, but for the Bradford team, actually recovering infectious virus from the salivary glands, I think we should be just kind of going back to that question again. Like, is it just Paraniscus that's important in maintaining this virus in natural populations? And going back to that level of evaluation for how these viruses are actually maintained. And they might be maintained in different ways. Like there might be different factors that contribute to prevalence in different areas. There are occasionally cases of Hantavirus pulmonary syndrome that are locally acquired, even in Michigan. And then they went sampling rodents around that household and found and recovered a virus very similar to the patient's virus from chipmunks and Paraniscus around their household. So it's not just localized to the west. I think that there are also going to be cases in the east. I do wonder how much humidity matters for that kind of translation from hazard, meaning the prevalence of the virus in rodent populations, to risk, meaning the realized human acquired cases of these viruses. And I bet that humidity matters a lot. And so if we have areas that had, maybe more humid summers, if they are transitioning into more dry summers, that's likely to facilitate more transmission and that you aerosolize more efficiently in dry weather than you do in humidity. So it's possible that the distribution of where you have risk to humans might be shifting with climate change as well in ways that we're necessarily anticipating. We also have a lot of open questions. Maybe this isn't what you're hoping for, is all the data gaps, but in terms of climate adaptations that people are making. In the Western United States, a lot of folks are converting to no-till. And so that makes more drought-resistant agriculture. That's something they've been doing in Argentina for a long time. So Argentina is already a no-till, largely no-till cropland system. And we're transitioning to no-till, and I don't know what that means for Hantavirus risk. Because on one hand, when you till, that's a major disturbance, right? And so the rodent communities are disturbed, and then they're moving into refugia. So like either you have tree stands typically lined up around agriculture for wind breaks or sound breaks, but then also rural homes. So if you go out there when they're tilling, you'll see hawks and birds of prey circling, and they're just waiting for all the rodents to start fleeing. And the rodents are fleeing either to the homes or to bushes, right? And so that's a big problem, we assume, in terms of that's a high risk time for people who live in these areas, is when they're tilling that might increase your risk for a short period of time. On the other hand, if you have no till, you're not tilling, you're not disturbing the rodent communities. Does that mean that you have larger rodent communities, that you're supporting a larger, more stable population of rodents? And I don't know the answer to that. And I think it's something that we really want to be able to study. But frankly, right now, there's just not a lot of funding to support that work. So it's still just a knowledge gap.
Well, friends, there you have it. That was part one. I hope you enjoyed it. As always, I enjoy sharing this information with you, and it was time to make something educational, with all the information that we have on social media and the news. So it was time for something like this. And please stay tuned next week for the second episode where we talk about the Andes virus, the cruise ship. So I hope you don't miss it. Okay, so next week is ASM Microbe in Washington, D.C. from June 4th through the 7th. So remember, if you see me there, say hello. I'm always looking forward to meeting you and connecting with you. And I have some great news. On June 6th at 3 p.m. on the ASM stage, For the first time ever, Let's Talk Micro is going to be having a live episode. So I'm so excited about this. So I hope that if you're attending, you stop by and show your support because this is such a first for Let's Talk Micro and I'm so happy about this. So please, if you're there, if you have time, stop by on June 6th at 3 p.m. at the ASM stage. And in this episode, I will be joined by Andrea Princey, which, as you know, we have done the AMR series before within the podcast. She's the co-host on that, so she will be co-hosting with me. And in the episode, we will be joined by Dr. Nathan Lidebor. And this is such a great episode, such a great topic. We'll be talking about whole genome sequencing and antimicrobial resistance. So please stop by on Saturday, June 6th at 3 p.m. Okay, so as always, you can find us on any podcast platform. So if you go to Apple, Spotify, Amazon Music, Goodpods, you can find us right there. So please subscribe to the podcast, download episodes, and if the app allows you to do so, please leave a review. Thank you so much for the support. And of course, you can find us on socials on Instagram, TikTok, YouTube, Blue Sky Social as Let's Talk Micro, X as Let's Talk Micro 1, LinkedIn as Luis Plaza. There's the website, letstalkmicro.com, and an email address, which is letstalkmicro@outlook.com. So either via email, via the website, via social media, you can reach out with any feedback, any topic suggestions. Those are always welcome and appreciated. And in the website, you can find information on study resources, licensing information, episode updates, and more. And as you know, this is a podcast that doesn't have any sponsors. So if you want to support us in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI, and both links are available on the show notes. Okay, my dear micro friends, as members of this community, either as microbiologists, infectious disease physicians, pharmacists, learning about antibiotics can be difficult. So if you want to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And as always, these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, micro friends, it's the end of this episode. I hope you enjoyed this first part of Hantaviruses. As always, I enjoy sharing this information with you. And hey, thank you so much for the support. I'm so grateful. And like I said, if you're at Microbe in Washington, D.C. from June 4th through the 7th and you see me, hey, say hello. I'm looking forward to meeting you and connecting with you. That's such a great event. So if you're attending, I hope you enjoy it because, I certainly always do. So, and hey, if you have the time, please check out the live episode at the ASM stage. I'm looking forward to seeing you there. So please continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.